This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Today, we're talking about a problem that immunologists ignored for nearly 100 years.
The dirty little secret was that all of these immunologists had been trying really hard to identify B-cell responses and T-cell responses. What people weren't talking about was that you can't take just foreign protein and inject it into a mouse and get any meaningful immune response. And so in order to study these things, they had a trick.
And we'll discuss the man that put that problem back in the spotlight. This is so well reasoned. It fits so well the data that was already known, and it happened to be correct. So when actual data started coming out that supported it, it was like, oh, everything we're discovering fits this paradigm. Finally, we'll discuss efforts to use our understanding of innate immunity to make better adjuvants and improve responses to our toughest vaccine challenges.
You have varied responses against flu vaccines. So some people, they make a protective response, and some people, it's not. There's this variety of phenotypes that you can study, whereas a lot of vaccines, like yellow fever, 1000% work.
Welcome to Audiommunity. Today we're going to be shifting gears a little bit away from the adaptive focus, but today we want to touch on something that I think is a little bit closer to what Kevin and Kate are working on, which is innate immunity. And
Woo! Innate immunity! Yeah!
So actually Kevin and Kate are both in a lab that is pretty directly, actually you're sort of derived from what we're going to be talking about today. You're like the great-grandchildren of it.
Yeah, I like to call Charles Janeway, who we're going to be talking about shortly, as my scientific grandfather. Great-grandfather.
Oh my God, Kevin.
I said it in my blog post. I called him my scientific great-grandfather in my blog post.
Yeah, it's too late. It can't be taken back.
It's too late. It's already out there.
Yeah, it can't be taken back. So
I would just like to go on the record and say I do not consider myself the great-grandchild of a genius. There's just a little too much weight on that mantle for me.
Come on. So by the end of the episode, we hope to convert Kate to wear her mantle appropriately as the great-grandchild of Charles Janeway, scientifically. All right. So before we get to Janeway, which we'll talk extensively about, and I have to say that I'm a little bit nervous about talking about the paper that we've chosen to discuss, which is a really critical work or piece of work that he wrote in 1989. I'm a little concerned because that paper was so important to the way that immunology was sort of rediscovered as a combination of both innate and adaptive immunity, which we'll talk a lot about today. So hopefully Kevin and Kate will give me a little bit of leniency here, because I'm sure that they could probably do a better job of introducing this than I will, but hopefully we'll give it a shot and see how it goes. So before we actually get to that paper, I think it's important to talk a little bit about what innate immunity actually is. We've talked a bit about adaptive immunity, we've talked about B cells, we've talked about antibody production at some point, but innate immunity is the other arm of the immune system that is involved not in the generation of antibodies, not in the generation of T cells, but sort of the rapid response that happens when your body first meets something that's foreign and pathogenic. And it turns out that the innate system, even though it doesn't give you B cells, it doesn't give you antibodies, is a really critical component of the immune system, and it's really important in making sure that everything is kept at bay while your adaptive system is coming online. And actually, I think Kevin would make the argument that your innate system is actually a lot more important than your adaptive system, although...
Well, I just want to say that line that you're drawing between the innate and the adaptive immune system, though I think most reviews that you read, most immunologists that you talk to would probably draw that line. It's not that clear, because you said the innate immune system doesn't give you antibodies, but as we're going to talk about, the innate immune system is critical to allow your adaptive immune system to function. And the adaptive immune system has critical inputs back into the cells that we normally consider part of the innate immune system. So I think it helps us conceptually to draw these stark lines and say, here's this one chunk of the immune system that is not specific, that's the rapid response, and here's this other chunk of the immune system that comes in later and is specific. But really, those two things are inextricably linked, and you can't really understand one without the other. And I think Janeway, in the paper we're about to talk about, points to that. But yeah, anyway.
No, I think it's a really good point, and I think that this is something that comes up a lot in immunology. And with humans in general, we really like boxes. Boxes are super easy. We can take pieces of data and say, this fits this paradigm, and so it fits into this box, and we just sort of call it good. And I think what we're learning more and more is that those boxes don't always quite fit, and by drawing the boxes, we make things easier to understand, but unfortunately, sometimes that makes us pretend like those boxes don't touch each other or aren't fluid. So hopefully we'll get at a little bit of that today, and the paper that you'll be reviewing a little bit later on is actually exactly that, where you're looking at why the innate inputs are so important into the adaptive system. And you're right, it turns out that one does not work without the other. So before we talk about the cellular components of the innate immune system, and there are lots of them, I think it's important at least to point out that when we're talking about innate immunity, we are at some level also talking about barrier function, which is incredibly important. We won't really be talking about it today, but I didn't want to just sort of gloss over that, right? Because you are in contact with the outside world all the time, and the vast majority of the reason that you're not sick all the time is because you have really well established barriers. The skin is a great example of that. And it's not just physical barriers. We're talking antimicrobial peptides. There are things on your skin that prevent bad bacteria from colonizing. All of these things are considered part of the innate immune system.
Things in your gut preventing
Yes.
colonization of bad bacteria. Actually, Kevin and I just went to a talk talking about this, how the innate immune system is helping like your gut keep a barrier function.
Oh yeah, cool.
Yeah, there's the innate immune system in the gut signals to produce antimicrobial peptides and a layer of mucus that sort of keeps a nice barrier between the niche of the gut and the epithelium. Is that what you're talking about, Kate?
Yeah. I thought that was kind of weird though, because it was unclear to me whether or not the innate immune system is actually signaling mucins, or why the mucus layer completely just disappeared, or if it was just colonization like destroyed it.
Yeah, I don't think they've actually shown. Whether mucins themselves are produced, but it's clear that there's some antimicrobial. I think maybe it's that the antimicrobial peptides that are produced by the innate immune system fill up that mucin layer and keep it stable. And if you don't have that, then the bacteria can sort of swim through it and get closer to the gut.
Well, the mucus layer is supposed to be just physically a deterrent. And then I guess like there's the idea of if your immune system is weakened, maybe you're just getting more through, and that you have more cell damage, so you have less mucus. But those pictures, very clear like mucus layer, and then without, what was it, Mighty 88? Completely just like no mucus, bacteria everywhere. It was really striking.
No, that's
Yeah,
really
that's
cool.
work by Laura Hooper from UT Southwest.
Which
Cool.
I could not stop thinking of how Sherlock talks about Mary Cooper, right? Is her name Mary Cooper? Anyways, but in my head I just kept saying like, Laura Hooper, in like Benedict Cumberbatch's voice.
That's fantastic. Well, maybe we can touch on some of that work. That actually sounds super interesting. We should go into that at some point. But yeah, so we're not going to be talking, unfortunately, today about most of the barrier function stuff, although, as you just heard, there's a lot of really cool immunology involved in barrier function. But the arm of the innate immune system that we are going to be talking about is the more cellular, what people think of traditionally as part of your immune system, I guess, white blood cells. And when I was reading this paper, I was trying to think, what did I think of when I thought of the immune system when I was, in high school or something like that. I went to a science fair, actually. I judged a science fair, which was super fun in Revere High School a couple days back. And I was trying to figure out exactly where my understanding of the immune system was while I was walking around and seeing posters and stuff. And I think really all I had a sense of was that there were phagocytes, because you always see macrophages going around chewing on stuff, right? That's I think a vivid image that people have of the immune system.
Phagocytes are cells that eat things.
Yeah, yep, and we'll talk a little bit about those in just a second. But the only other real thing I think I thought of was antibodies. I had a conception of white blood cells eating things and antibodies, and those were really my only two hints. And it's actually really interesting because that conception is are the two sides of this really big debate early on in immunology, which was, what is the immune system? How does it work? How do you keep pathogens from completely taking over your body all the time, right? And some of the first hints of that went way back into the mid-1800s or so. So there was a guy, Mechnikov, who was born in what is now the Ukraine, and he was a student of marine fauna, actually, and that'll become important later. But eventually he became sort of this comparative anatomy zoology guy at Odessa University in what is now the Ukraine. He was the one that first sort of discovered phagocytosis, and he was looking at starfish larvae, and he was showing that certain cells in the starfish, as it was developing, were capable of ingesting much larger things and breaking them down. And the reason that that was important was because he was taking a look at human white blood cells as well, and he was finding exactly the same thing. He was finding that some of these cells seemed to be eating larger things, clearing them, right? And that's where phagocytosis, right, eating cells, really comes from, is this guy Mechnikov. And that view actually wasn't really well received at the time that he presented it. People knew that there were white blood cells, but they were actually thinking at the time that bacteria were taking advantage of that system, and the bacteria was disseminating itself farther around the body by basically invading these cells and then just sort of getting carried along. So
it's worth noting that that actually does happen. There are several bacteria that are capable of infecting cells that are normally designed to eat and clear bugs. Some bugs are able to take advantage of those cells and sort of live inside these phagocytes. So things like Salmonella and I think Listeria and some other bugs are capable of living inside phagocytes, and they do so happily. But normally now we know that these cells do have clearing functions for a lot of microbes.
Yeah, and tuberculosis as well, which is a huge problem worldwide, does a pretty good job of evading phagocytosis breakdown, actually. So anyway, this guy presented this rather heretical idea that phagocytosis was important in immune function and important in keeping pathogens at bay. But it turned out, obviously, that he was correct, so the people that were detractors turned out to be a little bit wrong on that one. I did find something that was sort of fun on Mechnikov, and he was convinced that aging happened because of the toxic environment that bacteria were establishing in your gut. So he believed that by drinking lots of lactic acid, he could prevent those bacteria from aging him, basically. So Mechnikov every morning drank a whole bunch of sour milk in hopes that that he would live forever, I guess. It didn't work out.
And
but
that worked out really well.
Yeah, it didn't quite work out. But what is interesting about that, he was probably feeding... He wasn't totally wrong, actually. He was probably supporting the first probiotics program.
He was probably very regular.
Yeah, exactly. No, he probably was not terribly wrong in the health benefits of that, which is really weird, actually. But I still wouldn't recommend drinking sour milk because it's disgusting. And it's probably contaminated with something, so don't do that. But he did it. So this guy basically marks the start of this cellular immunity. So there was a camp of people that called themselves cellular immunologists. And like most camps worth noting, there was an opposing camp as well. And we've talked a humoralists. About the people that made up the opposing camp, but they were called humoralists. Actually, they called themselves humoralists, and that comes from...
That's not humorless.
No, humor.
Although they might have been humorless as well.
Probably. They were mostly German. So, am I allowed to say that? I think I am.
I'm keeping it in.
Good. So they were humoralists, as they were the founders and strong believers in what is known as humoral immunology. A humoral response is basically an antibody response. And so this is people like von Behring, Kitasato, is that how you say his name? Do you know?
Yeah, he was the guy in Ehrlich's lab, right?
Yeah, he was. And so they had established that if you immunized someone against diphtheria toxin or they had fought off diphtheria or something along those lines, you could take serum from that person and you could transfer it into another person, and that person would be protected. And we talked a little bit about that last time. And then, of course, there's Ehrlich himself, which he discovered antibodies. He basically established the idea that antibodies could distinguish self from non-self. And so you have these people that were sort of playing tug of war over what was important about immunology, what was actually important in fighting infection. And on one side you have the cellular people looking at phagocytosis, and on the other side you have people looking at antibodies. And by and large, the antibody people won that conversation, and they didn't realize that actually they were both right? We now know that both the cellular people... There was a really great quote from an editorial piece in Journal of Leukocyte Biology, actually, that talks about two blind old men, right? One handling the trunk of an elephant, the other looking at the tail, right? And disagreeing over what they were actually looking at. Really,
that's a common metaphor.
Yes.
And the elephant.
Yeah, I like it. I hadn't come across it before, so that's weird. Anyway, I liked it, but they turned out to both be right, and they were both really.
It's worth noting that Ehrlich and Metchnikoff shared the Nobel Prize in Physiology or Medicine in 1908. Metchnikoff for his discovery of phagocytosis, and Ehrlich for his discovery of antibodies. And that's really the last time that these two camps shared anything until the late 20th century.
Yeah, and we'll get there, but it did take a very long time. They did not really appreciate each other. And largely, I think the humoralists won that conversation. And the reason that they won, at least in the short term, was because the antibody phenomenon could explain a lot of the things that people were seeing with immunology. First of all, it certainly explained how you would transfer immunity by giving someone else's vaccinated serum. But it also explained things like anaphylaxis and serum sickness, and all of these things that people were seeing as immunological phenomenon could all of a sudden be explained by antibodies. And that, I think that difference, that tug of war, really was won at the time that this thing called clonal selection theory came about. So we talked a little bit last episode about the fact that you have an infinite diversity in your B-cell repertoire, and that was theoretically known, but no one really knew how that would come about. How do you actually respond to pathogens? How do you produce antibodies? Why is all of this happening? And clonal selection theory, this is Burnett that we're talking about now. Clonal selection theory, I think it's actually important to read the theory in its own words, because it's really sort of a central focus of a lot of this immunology. And the first part of the theory says that animals, specifically humans, contain numerous cells. Those cells are called lymphocytes. The second is each lymphocyte is responsive to one particular antigen by virtue of a very specific surface receptor. So in the case of B cells, you're talking about the B cell receptor, which will eventually be the antibody that gets released. So the third part of the postulate is that upon contacting its appropriate antigen, the lymphocyte is stimulated to proliferate, which is what clonal expansion is, and differentiate. So this is when the B cell sees the flu, sees HIV, and is specific for it. It will start expanding. And finally, the expanded clone is responsible for the secondary response, while the differentiated effector cells secrete antibody. And what that means basically is that you have this B cell population that's expanded. Some of them will turn into memory B cells, and we've got the life cycle of a B cell up if you want to take a look at that. Some of them will turn into memory cells, and others will turn into the actual boots on the ground, the effector cells. And so this theory of clonal selection really changed the way that people thought about immunology, and it turned out to be really important and really true. But it also had the bystander effect of really confirming the humoralist's view of the world, which is antibodies are super important and T cells are super important, and they are. Don't get me wrong, they are. But it really allowed adaptive immunity to dominate the conversation all the way from when they first discovered antibodies up to this really important paper that we're going to be talking about, which was only in 1989. So I was actually talking to Emily about this, and I said 1989, and she said, "You mean 1889?" And I said, no, like less than 30 years ago this happened. Really, there was a huge domination in the conversation in the field about the adaptive side of immunity, and not much attention was being paid to the innate side at all. So
yeah,
then comes...
it's great how like every innate talk starts like, this was only discovered 20 years ago. This field is still so very new. And like to me, and probably to Kevin, it's like this field has been established since around the time I was born. So it seems
Right.
like it's always been around to me. But then like you hear all these people like, before innate immunity, it was just magic.
Yeah, no, it's crazy. It's crazy. And it's clear that not a lot of people were even really thinking about it, right? It was just sort of off to the side, and people ignored it as best they could and tried to just explain everything on the clonal selection side, right?
Yeah, the idea of this clonal expansion and the allure of these receptors that are rearranging and that allows specific recognition. I think I might have mentioned this in the last podcast. That idea was what really got me excited about immunology in the first place, because it's so weird that you would cut up your genome and make these receptors that were unique to each individual cell. And that was a really cool idea. And I think the allure of that specific recognition and the ability to understand the mechanisms underlying that was really fascinating. And people were stuck on it for a really long time. But once that was explained, I feel like things had sort of stalled out a little bit. And Janeway sort of addresses this in Approaching the Asymptote.
Yeah, and that stall is actually why the paper is titled Approaching the Asymptote, right? It felt like immunology was wrapping itself up to a lot of people. The cells had been discovered, the lymphocytes had been discovered, the BCR, the B-cell receptor, was understood, although the specifics of it hadn't quite been worked out yet, but there was a clear path, I think, to understanding all of the obvious questions that were being answered. And so they had talked about T cells, and they had talked about MHC, because you can't have the T cell response without the MHC, so the MHC was studied heavily. But you got the feeling, or at least I get the sense looking back, that people were starting to sort of, everything's all good. We've got it all figured out, right?
Let me pull a quote from the paper. From what Janeway wrote. I believe it is safe to state that our understanding of immunological recognition is approaching some sort of asymptote, where future experiments are obvious, technically difficult to perform, and aim to achieve ever higher degrees of precision rather than revolutionary changes in understanding. And I think that sort of sums up the state of the field in 1989. And when you're thinking of just about immunological recognition in the adaptive immune system, so that statement 25 years later is still really accurate. A lot of new things have been discovered largely because of the insight from innate immunity that we've gotten in the last 25 years. But the idea that adaptive immunological recognition has just achieved higher precision rather than revolutionary change is still accurate.
Yeah, I think so. And there's a couple examples I can think of that I'm sure that we will talk about in future podcasts of where that might start to change just a little bit. I'm thinking specifically right now of NK cell memory, but I don't want to get into that right now.
Yeah, let's not start that.
Yeah, but
3-hour-long conversation now.
Yeah, no, that's a long one, but it is cool. It is cool. But I think that's actually a good, introduction to starting to talk about the paper itself. So Charles Janeway was a mass guy. Actually, he would have grown up pretty similar to where we're recording this podcast at the moment. He was a Harvard College guy, graduated from HMS, but he was mostly well known as a Yale faculty member. So he was appointed to Yale in 1977. And the reason I'm giving a little bit of background on Charles Janeway is because this guy quite literally wrote the book on immunology. If you have ever taken an immunology class, there's, I would say, a greater than 80% likelihood that the book that you used is called Janeway. So everybody just says, which version of Janeway do you have, right? That's how important this guy was in influencing the field.
Yeah, the actual title is immunobiology, but no one calls it that. Everyone just calls it Janeway.
Yeah, everyone calls it Janeway. And that's totally reasonable, by the way. Everybody accepts that that's reasonable. He was really, really influential. So in 1989, Janeway had been working a little bit off to the side with several other immunologists on this sort of heretical topic of innate immunity. It wasn't heretical, but just not getting a lot of attention, not getting a ton of funding, but a couple people were definitely working on it. So he was asked to introduce a Cold Spring Harbor symposia on immune recognition. That was in 1989, and this was actually going to be the third Cold Spring Harbor symposium on immunology. And I think there was one in 76 or something like that, and one in 64. So these things were pretty few and far between. So it was a pretty big honor, I think, to be asked to write the introduction. And if you actually if you're taking a look at this paper, and usually I would say if you like what we're talking about, you go see the paper, it's usually worth checking out. If you are interested in immunology, the paper is awesome. It's a really, really good read. It's pretty dense, but it gives you a great understanding of where the field was in 1989. He covers almost everything in it. And it's clear that he took it really, really seriously. He conferred with a whole lot of people in the field before really writing it. But part of the reason that he had to confer with so many people was because he also knew that by publishing this, he was sort of changing the way that people were going to think, or not. I mean, maybe there would be a huge backlash and he'd just get yelled at or something, but it turned out to be a really critical turning point in the immunological field. So sort of with that in mind, with the weight of it in mind, he begins the review with sort of the findings of the previous symposia and really just goes over what Kevin and Kate and I were just discussing, which was antibody structure had become really important. Everybody was trying to identify B-cell diversity. Everybody was trying to figure out T-cell receptors and MHC identification and all of these sorts of things. Random gene arrangement was coming into play a lot. And all of these things were really focused around this clonal selection hypothesis because immunologists just had it figured out. We had figured out diversity and we'd figured out how the immune system deals with the outside world. But after he closes the summary of where the field is now, he points out a pretty big problem with all of that. And this is where it gets really interesting. He calls it the Landsteinerian fallacy. And so Landstein was, and Landstein? I assume it's Landstein.
Yeah, no clue.
Is it EI or is it IE?
It's EI, Landstein.
It's Landstein.
Landstein. Okay, we'll stick with Landstein fallacy.
It's Frankenstein! Frankenstein! Sorry.
I like it. Let's keep that. So Landstein is one of the critical players in clonal theory. And one of the fallacy that Janeway points out is that one of the things that clonal selection theory assumed in just sort of being a standalone immunological function was that all macromolecular foreign antigen is created equal in its ability to stimulate an immune response. So it doesn't matter if you have HIV or influenza or steak, right? We all eat something that is foreign. We have these large foreign macromolecular antigens that are in contact with your immune system at any given moment. And one of the problems with pure clonal selection theory is that it would assume that the only way that you're identifying something as foreign is whether or not you've got a B cell that recognizes it. And that just doesn't work. It doesn't work because we see so many things. Our immune system is constantly inundated, especially at the gut. Gut immunology is becoming huge. It is constantly in flux as far as bacteria that are in there. You have huge amounts of bacteria in your gut. Anything that you eat that comes through, all of these things are foreign, and you have B cells against all of them. So the question is, why aren't you responding to every single foreign thing that comes through your intestinal tract?
And I'll say that most people at the time that Janeway was writing this sort of assumed that there's something called the, I don't know, Matt, if you were going to get to this, but there's the negative selection hypothesis, which is that B cells, as they're developing, if they come into contact with anything that they're specific for, they're going to be deleted from the repertoire. And this is true. This does happen. The assumption was that this accounted for self-non-self recognition, that any B cell that was specific for self or specific for something that you came into contact with, like dietary antigens, those B cells would be deleted from the repertoire during development. And that, it was assumed, was sufficient to explain self-non-self recognition.
Right. And then Janeway goes on to, and I know Kevin likes this phrase, and I actually asked some people if they had heard of it before. Most hadn't, but Janeway refers to immunologists' dirty little secret after talking about the Landsteinerian fallacy. And The dirty little secret was that all of these immunologists had been trying really hard to identify B-cell responses and T-cell responses. And so what you do is you inject some sort of foreign molecule, often...
Immunologist's favorite is ovalbumin, so albumin from chicken eggs.
Right, yeah, we really do like that. Although at the time they were using a lot of haptenated stuff because they could really track B-cell responses to haptenated proteins. We won't get into what that actually entails, but what people weren't talking about was that you couldn't actually just inject those haptens. You can't take just foreign protein and inject it into a mouse and get any meaningful immune response. And so in order to study these things, they had a trick, and everybody knew the trick, but no one really talked about it or why it was important, and that was the dirty little secret. And the trick was that you had to not only inject the protein you were trying to vaccinate against, you also had to inject something called complete Freund's adjuvant. And complete Freund's adjuvant is essentially just a smashed up MTB soup, right? So you take a bunch of tuberculosis, you mash it up, and you inject all of the parts, the really inflammatory parts of tuberculosis, alongside whatever you're trying to vaccinate against. And what that does is it essentially takes an innocuous antigen, that protein that you're trying to vaccinate against, and all of a sudden you create this huge B-cell response, huge T-cell response. And that's what people were using to try to identify how B and T-cell responses were developing. So Charlie Janeway, basically, in approaching the asymptote, points out that everyone is doing this. Everyone is injecting CFA or alum, which is currently an adjuvant that we use in our own vaccines, and pertussis, and all of these nasty, nasty things. But no one knows why. No one knows why you have to inject CFA, because if it's true that clonal selection, if the only reason B cells are going to respond is because they can respond, then they should be responding regardless of whether or not you have these other secondary things that you're injecting. And so this sort of brings into focus something called the second signal hypothesis, which he talks extensively about in this paper. And it's important to point out, I think, at this point, that he didn't have a ton of data supporting this at the time. A lot of this was based on some experimental findings and some observations here and there, but there wasn't a lot of data for what he was talking about. So he's essentially a priori just trying to theoretically work out how innate immunity must be working in conjunction with adaptive immunity. And what he comes up with is
it's
this...
kind of... Oh, sorry, Matt.
Go ahead.
Yeah, it's actually really hard from a modern context to describe this to students. So I've tried to teach about innate immunity to students before, and this idea of the second signal and the need for the second signal, it seems almost self-evident in modern immunology. It's such a fundamental part of our current understanding of how immune responses get triggered that it's really tough to explain to students that the theory that Janeway put together, yes, it was based on some experimental evidence, but the framework that he builds in this paper really was revolutionary. And it was not clear that he was correct when he was writing it. It wasn't an obvious thing. He sort of cobbled together a number of different ideas into this hypothesis. And like Matt said earlier, it really changed the whole structure of immunological theory to the point where it's hard to impress upon students how revolutionary it was, because in modern immunology, it seems almost self-evident.
It's funny because it almost seems like it was self-evident to him. And he writes this paper in a really forward-thinking way and lays out a really, really good evolutionary logical argument as to why innate immunity must actually be laid out this way. And what he's specifically talking about, and this is, I think, where we really need to get to in order to talk about the paper that we'll discuss, is this idea of pattern recognition. So we know that there is specific recognition of B cells, antibodies recognize specific things, but that didn't seem sufficient to totally make you understand what was self and what was non-self. So essentially what he was proposing was that you must have a more ancient, evolutionary conserved set of receptors that would allow you to distinguish as to whether a foreign protein or a foreign peptide or whatever it is dangerous and worth responding to. And so he actually coined the term pattern recognition receptors I think it was in this work. I think it came from this. And this is a term that we still use today. He also coined the term pathogen-associated molecular patterns. We talk about PAMPs all the time in modern immunology. So this really did actually define a lot of how we talk about innate immunity moving forward. And so what
I just want to say, so I'm in the process of writing my PhD dissertation right now, and it's obviously on innate immunity because that's what I study. But the theoretical framework that Janeway lays out in 1989 is the exact same, almost with no modification, theoretical framework that I start my dissertation in 2014. 25 years later, this hypothesis that Janeway put forward, really with almost no direct experimental evidence, is still the basis for understanding the theoretical understanding of innate immunity today.
And so these pathogen-associated molecular patterns, like I said, we call them PAMPs often. He lays out an argument as to why these things must necessarily exist, basically. He has no real evidence yet that they do. He has some thoughts. Actually, some of the thoughts turn out to be not quite right. What he thought was that T cells had these pattern recognition receptors and were going to see their MHC peptide that they were specific for, and at the same time receive a signal using one of their own pattern recognition receptors. And that would allow the T cell to determine, yes, I'm specific for this, and yes, this is something that's dangerous and should be responded to. That's close, actually. It turns out that most of the real decision-making happens in the antigen-presenting cell place. So these are innate immune cells that pick up some sort of pathogen for an antigen and then use those pattern recognition receptors to determine if the pathogen is dangerous. And so what actually is a pattern recognition receptor? Essentially, the framework that he lays out is that if the immune system is an evolutionary tool that is in constant battle with something foreign, then your immune system necessarily needs a way of identifying whether that foreign thing is there and whether or not it's dangerous. And so how do you do that? Essentially what he comes up with is this idea that pathogens often look like each other at some level, right? Just like mammals at some level look like each other, you could find distinguishing characteristics that identify all mammals. You can also find distinguishing characteristics that would allow you to separate different kinds of infectious material. A really good example of this is gram-negative bacteria. Gram-negative bacteria have a really thick outer cell wall, and that is really, really useful for them in living in the types of environments that they need to live in order to be pathogenic. So the cell wall is critical for the bacteria to function properly. What that means is that that bacteria is going to have a really hard time changing the way that that cell wall looks, because if they change it too much, the cell wall isn't going to work anymore, and so the bacteria is not going to be able to inhabit the same place, and it will die, or it will stop being infectious.
The analogy I like to use here is that if you want to detect cars generally, you can imagine there are red Ferraris and there are black SUVs and there are any number of different types of cars. But if your interest is in detecting cars, what are you going to look for as something that is somewhat unique to cars and will describe all of them, regardless of their individual shapes? And you could think about something like four wheels in roughly spaced proportions. Cars can't really change their wheels to squares. They can't really operate with, or they wouldn't operate as well with two wheels, although have motorcycles, but it's irrelevant for now.
Not cars. Not cars.
But
different.
Not cars.
Right.
So these pathogen-associated molecular patterns are parts of microbes that are essential to their function and very difficult to change over evolutionary time because their structures are vital to their behavior. So in the same way that you can't make car wheels squares, bacteria can't change the shape of their peptidoglycan in their cell walls.
Right. And so this is, like I said, all sort of based on theory when he lays this out. He doesn't actually know what these pattern recognition receptors are, or what they would look like, or even what cells they're necessarily working on. But he assumes that they must be there, they must be true, because of the way the immune system seems to work, because of adaptive immunity's dependence on another cell or some other signal that allows the adaptive immune system to tell whether something is worth responding to or not. And so as soon as he gave this talk, actually, Kevin, or Kate, does anyone know how well this was received at the time?
I was just going to ask that. Yeah.
My impression is that it was well received. It's like, oh yeah, that's interesting. But it didn't really fundamentally shift the way that most people were doing experiments. It didn't cause a bunch of people to all of a sudden be like, oh, I should start a lab based on these ideas. And that's evidenced by the fact that it took, I guess, 6 or 7 years before, I think Human Toll was 1996, I think.
Yeah, it was.
So 7 years later, Janeway publishes a paper that sort of starts, it identifies the first example. And it's not even a perfect demonstration, but I don't know, Matt, if you were to go into this history.
Yeah,
but
so,
in the mid-90s...
I was, yep.
Oh, okay.
Yep.
You're welcome to do.
Okay. Well,
so I guess the answer to your question is that people didn't think he was an idiot. People didn't say like, but people also didn't say like, oh my God, this really is a revolution. We should start studying it immediately. It was sort of like, that's nice. We're
right.
Going to go back to our T cell receptors now.
Right.
Until people started actually showing that this was true.
Yeah,
Sure.
but I would think as far as scientific revolutions go and like shifts and paradigms, that this one was pretty well received. Because, I mean, like, when the actual evidence came out, there was a lot more respect and a lot more, yeah, this is a thing, and more research. More people actually began researching in these topics. Whereas other more hotly debated things, I mean, I'm trying to think of a good one. But as far as, like, revolutions go, I feel like this one was pretty stable. I'm talking about this in a class I'm taking. Why are some advances in science... why are they like accepted by scientists and why are they not? And I was thinking maybe because of Janeway's conceptual layout of why this is important, that really paved the way. Once the research came in support, people thought, yeah, this makes sense that we would have it. I'll accept this and I'll research it, or when I review papers on this topic, I'll accept them.
Yeah, and I think also, I mean, like you said, Kate, this is so well reasoned. It fits so well the data that was already known, and it happened to be correct. So when actual data started coming out that supported it, it was like, oh, everything we're discovering fits this paradigm. And I think it also helps that he wasn't actually saying that anybody was, strictly speaking, wrong.
True.
Right.
I mean, the clonal selection theory that negative selection alone was sufficient, that was wrong. But he wasn't directly calling out current researchers as being wrong. He was basically adding a whole new chunk of theory that people hadn't considered before on top of all of the things that were true about clonal selection and about T cells and B cells and rearranged receptors. He was just saying, all that's good, there is also this other thing. And I think that helps because it was revolutionary, but it wasn't overturning previous dogma. It was adding to dogma.
It was adding a new layer almost. And you're right, it is, teleologically, it's extremely well reasoned.
Yes.
You know, it would be really hard to go through that paper and to point out logical flaws in the thinking. I'm sure you could do a couple things here and there, but it's really nicely done.
There are predictions that turned out to be wrong.
Right.
But only in like mechanism, not in like the idea of it. Like
absolutely.
The philosophy
conceptually,
of it.
Like I said, 25 years later, conceptually, I'm using the exact same framework for my thesis. So.
No, it's pretty impressive. Pretty impressive.
So I don't really like know all the history of the rest of immunology, but dendritic cells weren't appreciated to have the functions that they have that we know them to have now. Is that correct? He couldn't have predicted the function of a dendritic cell at this time anymore.
Actually, the data on dendritic cells at the time, and this is evident from what he writes, was basically that dendritic cells are constantly active, and they were constantly able to present to T cells and constantly, it was basically this hyperactive presenting cell.
Okay.
And so that didn't fit into his understanding because that wouldn't allow for much tuning of immune function.
Yeah.
And so he sort of had to put dendritic cells off to the side a little bit and say, well, I think that this is just a viral thing, or we now know that dendritic cells are incredibly responsive to these pathogen associated molecular patterns. So
and the reason that dendritic cells were always active in the context that experimenters were using them was an artifact of the way those experiments were done.
Yeah, exactly.
So dendritic cells looked like they were constantly active. We now know that in tissues, naive dendritic cells aren't capable of stimulating T cells. They're not capable of initiating an immune response until they get activated by PAMPs.
So let's talk about one specific pattern recognition receptor class, and those are the receptors that people normally talk about when they are talking about PRRs, and that is toll-like receptors. And so this is a class of receptors. Originally, they were identified, so toll, the reason it's called toll, or referred to as toll-like receptors, is this protein toll was identified originally in fruit flies, and it was identified completely independently of any sort of immune function, because back in I think it was the 30s, 40s, there was a lot of fly genetics going on. And what people were basically doing was irradiating these flies and getting horrifying fly mutants out on the other side. And the idea was that they were identifying proteins that were important in fly development. And it turns out that if you mutate this toll protein, these flies develop really abnormally. And so toll had been known for a long time, but it had been known as this developmentally important protein. However, that all changed in the mid-90s, and it changed because people started realizing that flies did have an immune system, but didn't really have any of the historical immune functions that we think about when we're talking about clonal selection. And so finally in 1996, Jules Hoffman demonstrated that these toll receptors in flies could actually prevent fungal infection. So if you knocked out toll, or you disrupted toll, these flies were really susceptible to this one specific fungal infection, demonstrating that somehow this toll receptor was interacting with the immune system, important for immune function.
Now, really fun with the toll stuff. You got to look at the picture of the fly with the fungus growing out of its eyes. It's really gross and really awesome.
I also want to say that the reason that it's called toll is because the woman in whose lab toll was initially discovered for its developmental function, Christine Nusslein-Volhart, is German, as her name suggests. Christiane Nusslein-Volhart. And toll in German means like cool. And when her grad student brought her this really, really screwed up developmentally fly, when he initially mutagenized it, she said, "Oh, that's toll. That's cool." And so that's why fly geneticists get all the best names.
Oh, they're super, super fun. I love fly genetics, actually. The names are awesome. They're really creative. I'm always impressed by that. Yeah. Worm people, too. Worm people are really creative. I feel
and plant people, too. It's just the mammalian geneticists that are stupid and rename awesome genes to things that are stupid.
Yeah. Although we do have Sonic Hedgehog, although that was discovered in flies.
Yep.
Damn it. All right. Well, anyway, we'll get back to that later. But getting back to toll-like receptors, so in 96, Hoffman found that toll was really important in fungal infection. And obviously at this point, Janeway and I think his still student at this point, Metztoff was the student?
Postdoc.
He was a postdoc. So Metztoff was a postdoc in the Janeway lab. They almost immediately then demonstrated that if you take an antibody that is reactive against toll receptor 4, so there are lots of different toll receptors out there. If you ligate toll receptor 4, we call it cross-linking, basically artificially bringing those receptors closer together, and the result of that is that you get signaling through that receptor.
Wasn't that done with fusion proteins in CD4?
Yeah, they didn't use antibody cross-linking in the first paper.
Oh, really?
They removed the ectodomain and replaced it with the ectodomain of CD4.
Okay.
And that caused spontaneous signaling.
Ah, got it. So, but basically what they were demonstrating was that if you do cross-link toll receptor 4, you could replace the effect of complete Freund's adjuvant, which I mentioned before, which is what everyone was injecting in order to stimulate adaptive immunity. Essentially, what they were showing was that just by stimulating TLR4 using this trick that they had, you could stimulate an entire downstream adaptive immune response, just like if you had added something like complete Freund's adjuvant.
Yeah, and I just want to point out, the logic of doing that experiment was actually quite interesting, because you have this fly geneticist that publishes this paper on fungal recognition in flies. And what Janeway and Metztoff realized is that the signaling pathway that in the cell that fly toll is activating is similar to a known signaling pathway in mammals that is involved in immunity. So they reasoned basically that here we have this receptor that's recognizing a pathogen, and it's activating a pathway in flies that is similar to a pathway in mammals that is known to turn on things like co-stimulatory molecules that can be used by T cells as a second signal. And they reasoned that there's this human homologue, this human protein that looks very similar to the fly homologue, and they actually called it human toll. It didn't get named TLR4 until a few years later. But they figured this human toll would have the same functions, and they showed, again, under Janeway's framework, that these receptors would activate something that would activate the adaptive immune system. They showed that this could, in fact, do that. It could turn on inflammatory cytokines, and it could turn on co-stimulatory molecules.
Right, and so this is really important, right? And ultimately, this sort of experimentation would lead to a Nobel Prize in this region. And the reason for that is this is the first demonstration that basically the whole postulate that Janeway had laid out was correct. There is this fundamental underlying assessment of foreign antigen that your immune system is presented with, and using these pattern recognition receptors, you can determine if it's something that's dangerous, and if it is dangerous, then your adaptive immune system will respond. So basically confirming everything that he laid out without any evidence. Now he's got evidence for it. And very soon after that as well, there were multiple people working on actually a bunch of different things surrounding toll. There was an animal model developed by Bruce Beutler's lab, and they would show that TLR4 was specifically sensing LPS. So the reason that that's important is we talked before about gram-negative bacteria. They have an extracellular cell wall that's really important to their function. And basically what Beutler showed was that the thing that TLR4 was sensing was a critical component of this bacterial cell wall.
It's actually the outer membrane.
Is it
Gram-negative bacteria have an inner membrane, a cell wall, and then an outer membrane.
Yeah.
LPS is part of that outer membrane of gram-negatives. Peptidoglycan is the
you're
PAMP
right.
that's part of the cell wall. That's recognized by Toll2.
Okay,
and
yeah,
some other
so
things.
In any case, we did have a pretty quick confirmation of what TLR4 was doing in an animal model, and this really establishes the idea that a lot of adaptive immunity is critically dependent on this pattern recognition signaling. And so that is really where I think the paper that you're going to be reviewing for us now kind of kicks off. That's the fundamental framework for it.
Before we do that, I just want to real quickly talk about, because you said that it was a mouse model developed in Beutler's lab, and I just want to be clear, so this was a well-known mutation in mice that made them insensitive to LPS. And the contribution of the Beutler lab was to do forward genetics on this mutation that had been known to make these mice insensitive to LPS. And it should be said that in the 90s when this was done, this was an incredibly laborious procedure. These days, you can sequence the entire mouse genome if you have enough money and a couple weeks, but back then it was an entire PhD thesis could be on sequencing a single gene. And so they basically identified where this mutation existed in the mouse genome, which was quite a feat. And it turned out to be at the TLR4 locus. And because the LPS receptor was at the TLR locus, which had been shown by Janeway and Metzetoff to be able to activate the adaptive immune system, those two findings together are really what cemented the TLRs as a pattern recognition receptor in Janeway framework of pattern recognition receptors, something that recognizes a fundamental component of a microbe, a pathogen-associated molecular pattern, and that is capable of activating the adaptive immune system through co-stimulation. That's all right.
Oh, and since this discovery, obviously this was really important. It received a Nobel Prize in 2011. Hoffman, the person that had discovered that toll was important in fungus. Ralph Steinman, who actually unfortunately died just prior to the release of the award. That's an interesting tidbit in Nobel Prize history if you want to go back and look at Ralph Steinman, but he had done a lot of the work looking at dendritic cells, which are one of the major antigen-presenting cells that take advantage of this TLR system, and Beutler for identifying that LPS was really or TLR4 was the LPS sensing mechanism. Those three things combined, like you said, in sort of the Janeway context, received the Nobel Prize in Physiology and Medicine in 2011. And since then, actually, since these discoveries, a huge repertoire of pattern recognition receptors have emerged. It's not just the toll system, although there are many tolls. They recognize a lot of different things, anything from DNA to RNA to flagella, which bacteria use to sort of swim around, to cell walls, and even some sensors that respond to cells dying. So you can imagine if you have an active viral infection, there's going to be a lot of cell death around, and your immune system can see pieces of your own dead cells and assume that there's a problem because there shouldn't be so much cell death in the local environment. And so all of these things have different receptors. They're all hard-coded into the genome. There are lots of different classes. It's not just tolls, but I think specifically we're going to be talking today about the tolls and what they respond to.
Yes, indeed.
So yeah, maybe that's a good starting point to jump into Bali Palundrin's paper.
Yeah, let's do that. And just to link it to what we were talking about in the last episode, we said last time that for a long time when making vaccines, scientists have sort of used a slapdash approach and basically figuring out what works, not necessarily how it works. And we've had a lot of success with that method. We've had a lot of really great vaccines being made. But as we mentioned last time, there are a lot of things that we don't have great vaccines for, despite a lot of effort at generating them. And so the context of this paper is to basically say, okay, we've been making these vaccines without knowing what we were trying to do. We were just basically throwing antigen into a system, maybe with an adjuvant of some sort like alum, which is the only adjuvant that's approved in the US. And sometimes it works, and often it doesn't. And so the idea of this paper is to take our current understanding of the innate immune system, the signals that activate immune recognition, and say, can we make a vaccine using TLR ligands?
Right, and different combinations of TLR ligands too, right? A couple people had been working on using individual ligands as vaccine adjuvants, and actually that's being pushed through Europe. There is now an adjuvant that is available in Europe based on TLR7 function. But I think, and you'll get into this certainly, what makes this really cool is the combinatorial aspect of this paper.
Right, and so in the years since the initial discovery of TLRs, it's become increasingly apparent that not only are TLRs and other pattern recognition receptors saying there's an infection, have a response against that, they're also saying what type of infection it is, because evolution has selected for receptors that are detecting certain types of pathogens, that have certain types of pathogen-associated molecular patterns. And the integration of signals from numerous different receptors can give some indication about what sort of pathogen we're fighting. So if it's a viral infection, you might want one type of immune response. If it's a bacterial infection, you might want some other type of immune response. And if we're trying to make vaccines against a bacterium, you could imagine that we'd want a different immune response than if we're making a vaccine against a virus. And you might even want a different vaccine if it's an extracellular bacterium or an intracellular bacterium, one that lives on the outside or the inside. You would want a different immune response. So we're still fairly far away from understanding exactly how all of those signals get integrated, but this paper is trying to say, okay, let's take a look at some different combinations of TLR ligands. I don't know if we've defined ligand yet. A ligand is the thing that a receptor binds to.
In the case of TLR4, it would be lipopolysaccharide, right?
Right. And so they're saying, can we give these cells a signal that looks like a particular type of infection and gear an immune response to what we want it to be? And so in this case, they're doing influenza vaccine. We know that we have influenza vaccine. Currently, the way that we vaccinate against influenza is we grow a ton of the virus, and we kill it and then inject the virus. And viruses contain pathogen-associated molecular patterns. I don't know, does the flu vaccine contain alum as well?
It has all sorts of things that it can have.
Yeah, there's a lot of different formulation, actually, that goes into
It's usually
flu.
the aluminum salt thing.
Alum.
Yeah, it's alum.
Okay.
It's alum. But there's
Okay.
also the adapted
mist?
flu, the flu mist, right, which is essentially just an attenuated version of flu itself. So
Right. So
I don't think that has alum in it. It would be strange to me if it did.
it wouldn't. That would hurt.
Yeah, it would hurt a lot.
So basically, currently we take dead flu virus and we inject that into your muscle with alum, which is an adjuvant. So it's one of these Immunologists' dirty little secret components, not nearly as nasty as complete Freund's adjuvant. That would give you a high fever and
Scarring and all
and
of
scarring
those
and all
things.
kinds of terrible things.
Sure.
So we use alum, and that helps boost the immune response. And flu vaccines work, not 100%, but flu vaccines work okay. They provide an immune response. They give you antibody protection. But the point of this paper is to say, can we improve that? Can we make it so that if you get vaccinated against a particular flu, you're not going to be 70% likely to make a productive immune response? If you're 70 years old, you're going to maybe make an immune response. They're saying, can we really boost this vaccine, make it really, really great at making neutralizing antibodies using the toll-like receptor ligands? And as Matt said, using combinations of them. And so the punchline of this paper, which, by the way, is called Programming the Magnitude and Persistence of Antibody Responses with Innate Immunity, the punchline of this paper in the end is that a combination of a TLR4 and a TLR7 ligand give you a really, really great immune response, whereas either one of those combinations on their own aren't as good. So they're making these little microbeads out of a plastic called PLGA, and these little microbeads are about the size of a virus, which is kind of cool. Trying to mimic a virus. I don't know if the size actually makes a huge difference, but these microbeads contain these TLR ligands. They don't use lipopolysaccharide. They use MPL, which I forget what that stands for.
It's just an analog. I mean, it doesn't really...
Yeah.
It looks very similar to LPS.
Oh, I know what it is.
But
it's monophospholipid A.
Yeah.
Usually it's abbreviated MPLA.
Is that generated? So one of the problems with LPS is, generating it. Basically, you've got a really dirty bacterial cell.
You can get really purified at LPS. It's just really, really inflammatory. So MPLA is basically the piece of LPS that triggers the receptor is the lipid A portion of lipopolysaccharide. And monophospholipid A is a version of that. And it's been shown previously that MPLA triggers the intracellular TLR4 response. So don't want to necessarily go into this now, but TLR4 signals from two locations in the cell, and it has different signaling pathways. One of them is really inflammatory, and one produces interferon. MPLA produces more interferon, basically. The balance is shifted towards that response. So, and actually MPLA is, I'm not sure if it's been fully approved as an adjuvant or if it's in the regulatory pipeline, but MPL is, it looks like it's going to be an adjuvant. So that's probably why they're using it. And I don't know if this R837 is similar. That might be the toll ligand that you're talking about, the TLR7 ligand.
Yeah, it's actually
approved in Europe.
Yeah, that is the one that is approved, I believe.
So the idea here is we're going to use these drugs that have some efficacy as adjuvants on their own. We're going to try to put them together. And they put them in these nanoparticles, and they're using ovalbumin, which I mentioned before is immunologists' favorite protein to try to immunize against.
Right. Well,
it has nothing to do with pathogens, but it's really well studied.
Well, the reason is that we have these really easy to use T cells that respond directly to ovalbumin. Right, so it's very easy to follow an immune response because all you have to do is take a mouse, put these T cells that are already going to respond to ovalbumin into the mouse as well.
But they don't actually use those in this paper.
Right. The reason that they use them, or that you use ovalbumin generally, is because we've used this T cell system so many times that it's become important for us to be able to track those T cells independently of genetic markers. And so you can make something called a tetramer that will specifically identify ovalbumin responding T cells. So we basically just have developed the tools so heavily around this ovalbumin system that it's just very, very easy to use it and respond and follow T cell responses.
Right. So the first thing that they do, basically, they're saying, okay, we think that good flu vaccines induce good antibodies. So we're going to look at the antibodies that are produced. If we use alum as an adjuvant, or if we use just the TLR4 ligand, or we use just the TLR7 ligand, or we use them in combination. And what they show is that the TLR ligands by themselves do okay. They do better than alum by a little bit. But if you put the two together, if you use both TOL4 and TOL7 ligands, you use both MPL and R837, and you immunize these mice against ova, you get these huge antibody responses, and you produce a number of different types of antibodies, different IgG isoforms. We're not going to go into what those mean, but basically really good antibodies that are normally associated with neutralization in serum. And you get those after a single dose, and you also get even more when you give a booster shot, essentially. And in addition, they go and they test.
And did you know, Kevin, do you know if they if they tested, so you have one nanoparticle is what these things are called, these beads. You have one nanoparticle that has both MPL, so the TLR4 agonist, as well as the TLR7 agonist. Did they test what happens if you inject two different nanoparticles, one that has MPL and one that has R837, or do they have to be in the same nanoparticle at the same time?
I don't know. I didn't.
Well, they do the separate, the R837 or whatever, and the MPL separately with the HA.
Right, but I think Matt's saying if you did like an HA nanoparticle, a TLR4 nanoparticle, and a TLR7 nanoparticle as like three separate rather
I see
than.
What you mean.
Yeah, I wonder.
I don't know the answer to that. I do know that some other papers have shown that if the same endosome, so the same compartment of an individual cell, has both a TLR ligand and the antigen, the protein antigen, it'll be much better at stimulating a T cell response than if the protein is fed on one side of the cell and the TLR ligand is fed on the other end of the cell.
So maybe it's important, maybe that these things are in the same place at the same time. Potentially.
Yeah, ultimately, I mean, I think you'd probably get the best effect if you had a single bead that had both toll ligands and the protein on it.
Okay.
I don't know why they didn't do that, if that's technically not possible.
Yeah, I mean, I've made nanoparticles before, and it's a pain to assess what they actually are.
Okay.
So I can tell you, it's actually really easy to make the particles themselves. It's just a series of sonication steps. But actually determining what you have once you're done is really, really difficult.
Yeah, it looks like it's just easier to make them than
Yeah.
to
Yeah.
like put them in. And I think, I don't know.
It also makes sure that the dose of your it makes sure that the dose of the antigen is consistent, because otherwise
Right.
somebody might complain like, oh, well, maybe this particle had more of your antigen.
Right, and that's what I mean. Like assessing on an individual particle by particle basis exactly how much TLR adjuvant you have, exactly how much antigen you have, all of those things is just technically challenging. That being said, I'm sure that they had the capability of doing that here. This is a really well-funded, really strong lab, so I imagine they just aren't showing the experiment. I'm sure that they've done it.
So the other thing that they do in the first figure, which I think is super important and super interesting, is most papers of this sort will stop at how much specific antibody did you produce. So you make a lot of antibody of a particular isotype that's specific for your antigen. But as we mentioned last week, during an immune response, B cells undergo something called affinity maturation, where over time they're going to cause mutations in the portion of the receptor that binds to antigen, and the ones that are better at binding are going to be positively selected for. And so, in addition to the sheer number of antibodies, you could also have differences in how strongly those antibodies bind to the antigen, and that's called the avidity. And in this paper, they go an extra step and actually test the avidity. Go ahead, Kate.
When you vaccinate, you're not inducing like the hypermutation or anything like that.
Yeah.
No, sure you are.
Really?
You get germinal center
absolutely.
responses.
I just listened to a vaccine talk, and they were like, of course we are not saying that vaccine increases the avidity of these antibodies. Like, literally, he said that.
Right, but so any time you get an immune response that generates IgG, so isotype switching, during that process of B cell maturation, you're getting affinity maturation as well.
Really?
So in germinal centers, you're getting... So a vaccination isn't necessarily going to give you better avidity antibodies than a natural infection would. Maybe that's what he was saying?
Yeah, maybe.
But basically, the only time you don't get affinity maturation is if you have a really, really bad immune response.
Or actually, so there are T-dependent and T-independent responses, right, to B cells, and we don't have to go into them heavily, but basically B cells do need T cells at some level to be effective, and so there are different kinds of sugar-heavy antigens and things like this where you can get the production of antibody, but you don't go through this affinity maturation process. But that's a really, really specific set of antigens that happens
it
for.
might actually be then specific to HIV-ONF.
It could very well be. Yeah, I'm not sure.
Someone also told me that, like, unless HIV-ONF is evolving and mutating that you don't get affinity maturation of those antibodies.
Interesting. Yeah, that I'm
Doesn't
not
make any sense to me.
That doesn't make sense to me either. But I could just not know anything.
Yeah, I was actually hoping that you guys could explain that to me, so because it made no sense to me too. I was like, what? Why do you need evolution?
It must be context dependent, or they're not talking about the same thing.
Well, HIV-ONF is covered in glycan, so it's covered in sugars, and it completely blocks a lot of antibody binding. So it's kind of a weird antigen to begin with. So it might just be weird HIV data.
Well, if it's really if it's a repeating structure on the outside and it's just huge amounts of repeats, then it could potentially trigger this sort of T-independent response, and then you're going to have a much harder time. But that being said, I would still be surprised if at some level T cells weren't gaining access to it. But maybe that's true. Maybe the T-independent response is just so strong that you don't get affinity maturation. I'm not sure.
It might be one of those things where like people know it and don't publish it or talk about it.
Right.
Interesting. If it is published, we should do that on a future episode where we'll talk about B1 cells.
Yeah.
But
yeah.
Not this time.
Yeah.
This time we're talking about normal T-dependent B-cell responses, and you do get affinity maturation. And what they show, which I think is kind of cool in this paper, is they show that not only are they producing more antibody, the antibody they are producing has a higher avidity. It's stronger at binding to the ovalbumin antigen. And they show this using a technique called surface plasmon resonance. And basically this is you affix the antibody protein, or you affix, in this case, I think the antigen itself, the ova, to a chip, and then you flow the antibody over, and the tighter it binds, the more electrical conductivity you get, I think, and that tells you that you have stronger binding or tighter binding, because those signals that they change for a longer period when the thing binds longer. So it's a well understood technique, and they show that basically these antibodies bind more tightly, which is cool.
I feel like it's a well understood technique that we do not well understand, unfortunately.
Yeah, well, it's like
it is well understood by someone.
Someone really knows how it works.
Well,
it's
it's like a relationship, right, between you have the flow channel and a chip facing into the flow channel, and the antibodies bind the antigen, and then you shine light in, and then the light goes in and refracts, and so that's correlated to amount of binding occurring. So
that makes sense.
More like kind of like similar-ish to measurements that you take where you look for density of something, like the OD measurements. It's like similar-ish. But the nice thing is you can watch this happen over time. So you just keep shooting in light, you measure the reflected light correlating to like the amount of binding that's occurring, and then over time as binding starts to dissociate, you see the antigen and the antibody lose the binding. And now once you have that sort of data, you can basically, like the full binding profile. So that's why SPR is great. So you you don't see like how quickly something binds the antibody and how quickly it unbinds.
Right. So the on and off rate is the biochemical terms.
Cool. Yeah, I did not know that.
I did not know that either.
Well, if you don't, so basically if you don't have the full curve of the off, you don't know anything. So SPR is really great for that.
Okay.
Yeah. Unfortunately, these things have to be dissociated from membrane. So people often use this technique to look at things that are normally membrane associated, and it's a little bit iffy if that's true. But in this case, antibodies are secreted and the ova is just bound. So this doesn't necessarily tell you exactly what it is, what the binding is in vivo under normal circumstances, and some people will try to make claims about how tightly it actually binds based on this technique. In this particular case, for this particular paper, they're looking at relative avidity of things that are normally secreted, and so this is perfectly acceptable, I think, in this case, and really tells you a lot that the antibodies, that the take home here is that the antibodies produced by having a combination of TLR ligands, not only are there more of them, but the ones that there are bind more tightly to their antigen, which is really important.
So you have more and better.
Yes. So the next thing that they do is they want to sort of establish some of the genetics, and they want to say that the requirements here are actually for the toll-like receptors that we think they're for. And interestingly, they are important in the B cells. So the way that they do this is they take mice that are deficient for one toll-like receptor or the other, or for adapter proteins, which we're not going to go into. MyD88 and TRIF, I sort of mentioned them briefly, but basically these are things that are required for TLR signaling. they take these B cells and they add them into mice that have no B cells of their own. So these are called mu-MT mice. They lack a gene that is required for producing their own B cells. And so they add in these B cells that are knocked out for various things and then look what happens in the mice. And the interesting thing they do also is that they include B cells that are knocked out for TOL4 or TOL7, and they put them into the same mouse. So they're trying to say, do B cells A, do they require TOL4 and or TOL7? And in addition, do they require those receptors are in the exact same B cell? And what they find is, yes, it actually is important for B cells to have both TOL4 and TOL7, which suggests that the action of this adjuvant, the reason that it works is because it's working on the receptors of the B cells. And this was actually a little bit weird to me because they have they have basically no antibody production if you knock out either TOL7 or TOL4 in B cells. And the same is true if you knock out either MyD88 or TRIF. And the reason this is strange is because even though I said, so MPLA is known to act more strongly on TRIF, which is the interferon producing part of the TOL4 pathway.
Is it killing you not to be able to talk about cascade signals on MyD88? Do you have?
Yes, a little bit.
So Kevin is writing a dissertation on the signaling pathway as we speak. This is something he's thinking about 24 hours a day. So
yes.
Yeah. Anyway, I figured that probably
it hurts
was killing
a lot.
you.
But the main thing that I want to point out here is that there's data from our lab, actually, that B cells that respond to at least lipopolysaccharide, which is another TLR4 ligand, don't actually get internalized and don't signal through TRIF. So it's weird to me that this pathway that we have data suggesting that doesn't happen in B cells.
When did we start working on B cells?
Well, Ivan's paper, the CD14 paper.
Oh, Ivan doesn't count. Ivan's too good for us.
It's from our lab. I didn't say it was we that studied it. I said it was our lab. But it's possible. So that same paper, they showed that paper was about this factor that pulls the receptor inside the cell. They also showed that if the particle itself could get inside cells by itself, that could bypass this need. I don't know if maybe that's happening here.
Yeah, maybe that's what's going on.
Because these particles probably can be endocytosed by these B cells, especially if they have protein ligands. Although the beads that have the TLR ligands and the ones that have the antigen are different. I don't know. It's a little confusing. In any case, it seems that the B cells themselves must be able to respond to the TLR ligands themselves.
Right. So maybe that answers my question.
Effect.
Right. Maybe that answers my question before about whether injecting two different nanoparticles at the same time would be effective. And this would suggest probably that it wouldn't be as effective because you'd want the same B cell to get hit with both receptors at the same time. So
Right.
you'd either have to saturate the environment where every B cell gets hit with everything or just hope that accidentally,
Okay,
Right.
here's a dumb question. Why do the B cells matter?
in the...
Yeah, so I understand that like B cells make antibody, but like B
Yeah.
cells can make antibody. Why do they need TLRs to make antibody? Because my virology point of view is like B cells interact with T cells and dendritic cells, and they tell the B cell to start making antibody. And I thought those signals were independent of TLRs.
So they are,
They
but,
generally are.
But there is a critical... So actually it gets back to our question before about T-dependent versus independent responses.
So
And in T-dependent responses, the B cell is talking to the T cell, and it's actually serving as an antigen-presenting cell in that scenario. So the B cell itself will be activating the T cell, and depending on how much signal the B cell got from the TLR response, it's going to be able to activate those T cells better, and so it'll all feed back. So basically, the quality of the B cell response can be influenced by the amount of TLR ligation.
So is
And there's
that like
actually work
known
showing-
for this immunization? That's the way that these was it like a shot in the dark to do this experiment with B cells, or was it generally known that B cells and like their TLRs are really important for this sort of vaccination?
I don't think it was a shot in the dark entirely, although it's a little surprising to me, but people have shown that, for instance, in lupus, if you have B cells that are specific for DNA, B cells can internalize DNA, which then triggers TLR9, which normally recognizes DNA, and that can activate B cells, including getting them to have germinal center responses, including affinity maturation, in the absence of T cells. So if you have a B cell that is getting a whopping dose of TLR ligands, if it is antigen specific, it can not only do what Matt was talking about, where it becomes an antigen presenting cell itself and can sort of get better signals from T cells, but they can also be activated in the absence of T cells entirely.
Yeah,
I didn't think that would be important in vaccination, but the data here is pretty conclusive, I think.
Yeah. And I think, you mentioned the lupus model. We look at that model directly in the lab, and I can tell you that the B cell activation outcomes are heavily influenced by their TLR repertoire on the surface.
Interesting. I never knew that.
Well, so now they know that this combination of TLR ligands is making lots of antibody, and it's making really good antibody, and they showed some of the genetics. It definitely depends on the TLRs, as they suspected. And so the next thing they want to do is they want to look at the immunization response over time. They look at the number of germinal centers. So the germinal centers are the location in a lymph node, which is where B cell activation is happening. And a germinal center is sort of this ball of rapidly dividing B cells that are undergoing class switching and affinity maturation. And they see that with this combination of both toll ligands in the nanoparticles, they get much larger and more sustained germinal center responses, and that correlates with more antibody production at those time points. And what they're doing in the last part of figure 3 is they're looking at basically the amount of specific antibody present in serum in these mice very, very long after immunization. For example, at 80 days, so almost 3 months after the initial immunization, the mice that received just the Toll-4 ligand, MPL, and ovalbumin, they're looking at this, the readout is number of ELISpots per million cells. So ELISpots is just a way of detecting specific antibody. So the number per million cells looks like about 60 in the ones that only got a TLR4 ligand. In cells that got both TLR4 and Toll-7 ligand, it's closer to 1,000. So 50 to 1,000, so much better. That's 90 days after. They take this experiment out really long, 550 days. So what's that, a year and a half, roughly? And at 550 days, basically none of the mice, other than the ones that got the dual treatment, have any antibody left in their serum. Whereas the ones that got both have about 150 of these ELISpots per million cells. So only the mice that are getting both of these ligands at the same time are having a response. And this is including alum, which is what we typically use for vaccination. Although weirdly, alum at this late time point seems to be doing better than any of the other treatments alone. So if you only have Toll-4 or only Toll-7, alum seems to be doing better at this late time point, whereas early it doesn't look like it's doing any better. But certainly the Toll-4 and Toll-7 ligands together are giving much, much better responses.
Yeah, alum is super interesting to me because I feel like alum, it just happened to work out. He sort of mentioned that we've stumbled through a lot of vaccine design, and for a while in the field, everyone thought that the response to alum was the result of this thing called the inflammasome, and it turns out not to be the case. And at the end of the day, at least as of, a year ago, right around this time, it was pretty clear at this conference that I went to that people really don't know exactly how alum is working. There are now some good thoughts on it, but basically the way that everyone thought it was working, it wasn't the case. So this is an adjuvant that's been really traditionally used, and we don't quite know what's happening.
Oh, how weird. I just read something from NIAID or whatever. I can't. NIAID?
That's it.
NIAID?
Yeah.
There we go.
Yep.
National Institutes for Allergy and National Institute for Allergy and Infectious Disease.
Yeah, and so they list all the different adjuvants that are currently under clinical trials and which ones have been approved in the U.S. and how they work. And it says aluminum salts, inflammasome.
Yeah. So it turns out that if you knock out critical components of the inflammasome, you still get adjuvant effect of alum. So
it's okay. Inflammasomes are made up. We all know.
Yeah,
this.
Right.
Not really, folks. Maybe.
Yeah.
Maybe.
No,
to be determined.
Yeah, everybody you discover something new and everybody gets really excited about it. No, it is important in certain things, certainly, but it turns out that alum, probably the primary mechanism that it's working is not actually inflammasome activation.
How weird.
Cool.
Yeah.
So speaking of alum, figure 4, they don't use it, and that's one of my main criticisms of this paper, but what they're doing in figure 4 is they're basically taking the stuff that they figured out in the first part of the paper, which has all been using this chicken egg protein that immunologists really love, but that has no relevance for infectious disease. And now they want to say, okay, what happens if we actually vaccinate animals against something important like flu? And
which is
so what they're doing.
My favorite vaccine model is flu. It's
Oh, yeah?
the only one
Why is
that
that?
makes sense. Because flu does not have flu vaccine is not 100% effective. So it's
Right.
60 to 80 depending on what the actual vaccine is. So you have a great, and everyone gets flu vaccine. So like to find samples of people who have gotten flu vaccine is not very hard. So it's like an easier system. In the UK, you can pay people to just be exposed to flu and do direct tests. So that's kind of cool too.
I want to know how much you get paid for that. I would potentially do that.
Probably not enough, but
Yeah, no.
But the great thing is that you have varied responses against flu vaccine. So some people, they make a protective response, and some people, it's not. So there's this variety of phenotypes that you can study, whereas a lot of vaccines, like yellow fever, 1000% works. It's gonna work, the end. And while it's like a great model, there's only like so much information that you're actually gonna get from it.
Sure.
Anyway.
It's sort of saturated on one end, you're saying?
Yeah. So I just I love flu vaccine studies. That was my spiel.
That was a good spiel. I liked it.
The virologist, everyone. I totally had.
Were you gonna continue? Sorry. I didn't mean to derail everything. You were saying.
No, I just wanted to say, so at the beginning we were talking about how Kate felt like Matt and I sort of dismissed her comments.
I didn't say dismissed. I said you just talked over me.
Oh, okay. Well, but I feel like it feels a little bit like I'm dismissing that comment, but really it's just that I have nothing to add because it's
just so
it's a very
genius.
compelling argument.
It's just so incredibly genius. And I already won Matt over with flu vaccine is the best thing to study.
Yeah, right.
True.
Nailed
That's
it.
I love her.
Yeah.
She's the greatest. So what they're looking at in figure 4 is what happens when we actually do flu vaccination. And so they're using whole inactivated virus. So this is what I mentioned before, is just basically take a lot of flu, you kill it, and you inject that in. And they're comparing a ton of whole inactivated virus to a little bit of whole inactivated virus to a little bit of whole inactivated virus plus these microbeads with the TLR ligands. And what they see is that if you include these TLR ligands, even with a lower dose of virus, you get much better responses than the inactivated virus alone. And that's cool, and the data looks really good. You get really great responses, even very early in the infection or in the immunization. But there's a couple problems I have with this. First is they show what happens with a boost at 35 days after, which for flu doesn't make any sense because right now nobody gets booster shots for flu. Maybe we should. It would probably improve things, but nobody does it, and it would be nice to see what these look like.
No. Immunosuppressed people will constantly get flu vaccine over the course of flu season.
Oh, really?
Mm-hmm.
Oh, I didn't realize that.
Yeah.
Yep.
Oh, yeah.
Yeah,
They'll
they
get
get
multiple
re-injected.
vaccinations.
Okay. It would still be nice to see.
It's just for most
what
people,
happens.
it's fine just to have the one, I guess.
Yeah. So you're looking for persistence of these antibodies, persistence of response without boost.
Yeah, that's one thing I'm looking for. And the other thing that I think would really, really be important in this figure is what it looks like with alum, which is what we currently use. And the reason for that is because as opposed to the chicken egg protein, which they showed earlier, whole inactivated virus itself contains TLR ligands. So flu has RNA, it's an RNA virus, and that RNA is a TLR7 ligand. And so whole inactivated flu alone, you do get antibody production. And it's probably because whole inactivated virus itself has TLR ligands. And it would be really nice to compare what our current vaccination strategy looks like compared to this vaccination strategy. When they're looking at this chicken egg protein, ovalbumin, they showed the comparison, but it's not the same situation because ovalbumin by itself is not immunogenic. Virus itself is immunogenic. So it would be nice to see what flu with alum looks like. At the same time, if you look at the titers of neutralizing antibody with this treatment that has both the Tol-7 and Tol-4 adjuvant in it, it's plotted on a log scale. So you have significantly more neutralizing antibody, but they also don't show the single TLR ligands by themselves.
Yeah, I agree with your first assessment, actually. I think that it would be really important to show alum, and I work with flu vaccination all the time, and we just we UV inactivate the virus and we just inject it, and you do get long-term antibody responses. So I think that's completely valid. I do think, though, that they, leading up to Figure 4, they show enough times that the combination of both is better, that I actually don't have a huge problem with them not showing the individuals here, especially in macaques.
And especially in a monkey model.
Exactly.
Yeah, they're doing this in monkeys, and those are really expensive, and ethically, it's harder to justify using a lot of monkeys compared to mice for whatever reason. I mean, some people might have an objection to that characterization, but that's generally true. Generally, people give more leeway to using less monkeys in a study.
Definitely.
Right.
Definitely.
It seems weird that they used 2 different TLR7 ligands in this figure and nowhere else.
Yeah, maybe they ran out. Maybe it was accidental.
and R837.
Yeah, it was probably the overly honest methods. We accidentally had an undergrad that was making these particles and used the wrong TLR7 agonist, but it was in macaques, so we just stayed with it.
That's possible, I suppose.
I would really like that, actually.
But I think, though I agree with you that maybe it's okay to not use both, although, like I said, because flu already has a TLR7 ligand intrinsically, I think it'd be different here. But I think leaving out alum, which is the current standard, it's really an oversight here, because the argument that they're making here is that these adjuvants are far better than what we currently have. And I think that's probably true.
Yeah.
But if you want to make that claim, you have to show in a real immunization model with a real vaccine target that it actually is better.
Yeah.
So.
Yeah. So it's still in mice, right? In the third figure.
Yeah.
Where they compare ELISpot data. Yeah.
Mm-hmm.
Yeah. So you've got indications, but you're right. I think if you're wrapping up Figure 4, it would have been really nice to have the direct comparison.
Yeah, and like I said, they use 2 different TLR7 ligands, which they don't use anywhere else in the paper, so it would make much more sense for one of those bars to be virus plus alum.
I agree. I think it's just a monkey issue, because they are so expensive. It's so rare to have giant monkey groups. And I'm like, personally, I'm fine with that. I think that we should use monkeys sparingly when possible.
Sure.
Yeah, that's definitely true. But I feel like not doing that experiment means that experiment has to be done by someone else at some future time anyway. Because you really need the side-by-side comparison with what's going on now and with what they're proposing in order to make this compelling, I think.
Yeah. No, I think I agree.
At the same time, I think this is a really cool paper showing that I would argue that this is using an almost 1990s understanding of the innate immune system.
It really is, yeah.
Which is great because it's better than the 1950s understanding that most vaccines currently use. But
sure.
But we're moving towards a time when our understanding of the innate immune system, the initiation of an adaptive immune response, our knowledge of that is going to inform how we generate vaccines. And I think that that's really important because right now, like I said, most vaccines up until very recently have been based on, let's just try it and see that it works. And hey, it's worked for the last 50 years, so let's keep doing it. And we really need, especially for a lot of these hard to crack pathogens, we really need a more up-to-date understanding of how these immune responses are being generated in order to make better vaccines.
So why do you think there are no TLR adjuvants approved yet?
So there is in Europe.
Yeah.
So the TLR7 is in Europe. I think
MPLA, is...
It's coming. I think the process is just really slow. The problem is that, and this is a problem with vaccination perception in general, and that is that these adjuvants, these ligands, are inherently heavily inflammatory. So if you inject someone with just pure TLR ligands, they have a really, really bad response. You just have to be really careful in the amount you're injecting. And you can see that. The problem is that there's a reciprocal relationship between the amount of inflammation and the amount of efficacy. So, it's not reciprocal, it's direct.
Directly correlated.
So if you cause a huge amount of inflammation, like the old smallpox vaccines, right? Everyone who got that vaccine has a scar on their arm. And it was a really nasty inflammatory response, but it also worked really well because your immune system's like, holy shit, guys, this is actually something problematic and we should make some really good antibodies against it. And so we play this fun game, I think, as an immunological community, like telling people, oh yeah, no, the vaccine's great, it's okay. And really what we're thinking is, I want to give you so much LPS right now and you will totally be great forever, but you're never going to get a vaccine ever again. So I think that we're sort of like doing this dance, and so it takes us a very long time to get these things through the pipeline.
Another component, I think, is that for the vaccines that work, it's unethical to withhold those vaccines and test something else that you don't know whether it works, right? You can't give someone an experimental pertussis vaccine instead of the known pertussis vaccine and see which one's better. That's not ethical.
Really?
I think your point...
Even if
absolutely.
It doesn't kill you?
Yeah, because then you may or may not be protected from pertussis.
Right, the thing is that, though, you could inject someone, theoretically, with a random... You could use ova in a human system if you were just trying to look at adjuvant efficacy. Because really, we're talking about getting the material approved for use in humans, not the actual vaccines themselves, because alum is the only material that we can use. But I could imagine a human study where you just use ova, right? You do the same thing that we do in mice and say, is this adjuvant effective? And then start to build a case for why you should switch over the pertussis vaccine.
Yeah, but again, my objection with figure 4 is going to apply to humans too. You're going to say, well, I don't believe this unless you show me the comparison to... And I think actually Kate's point about flu vaccination being a good model because it's not 100% effective. We know that normal flu vaccine is only 70% effective. What if you gave people the flu vaccine, as we currently do, you gave 100 people that flu vaccine, but you also gave 50 people, I mean, obviously the number should be higher for a real clinical trial, but you also gave half of those people, in addition, you gave them these TLR ligands.
Sure.
And then you see, does that 70 to 80% become 95%?
Yeah.
And if that's the case, then you could make the argument, okay, we know this improves things, let's see what happens on its own. And then you could do that direct comparison.
Yeah, I think you could come up with studies once the materials themselves are approved. I agree with you. I think you could work it in. The bottleneck right now, though, seems to be the FDA won't even allow you to test these things in humans, which I think is sort of stalling the front end of the development of these sorts of things. So, in any case, it looks a whole lot like Janeway thought it was going to look in the beginning, doesn't it? It's sort of his legacy come true, so I'm happy to see that for him.
Me too, because he's my scientific great-grandfather.
Still not comfortable
so Kate,
with that.
how close were we? Were we, like, at all close?
No, it just...
No, Kate just thinks that she's being pretentious by not... Or Kate thinks she's avoiding pretension by saying that.
No, I feel
really
like there's no saving me. Like, granted, I think a lot of people could say I'm a very pretentious person. I'm fine with that. But I have like that complex where like I'm not going to go around telling other people that I'm super hot. And that's just asking to be proven wrong. That's just asking for people to like completely disprove all of your papers.
All right, well, I think on that note we should probably wrap it up.
This has been Audiommunity from immunity.org. I'm Matt with Kevin and Kate. You can find us on iTunes now. You can also find us on Google Plus and Twitter. We're on Facebook at facebook.com/Audiommunity. And you can find all those links on our website, immunity.org. That's EMMUNITY.org. As always, we're supported in part by Harvard Medical School, and our music in the intro and the outro is composed by Rachel Rennick. Thanks for listening.